dehydrogenase complex after the glycolysis process. So we said the pathway, right? If the oxygen persists, the pyruvate is going to go to the matrix of the mitochondria from the cytoplasm. Okay, so pyruvate is still in the cytoplasm. So pyruvate dehydrogenase complex, this PDC stands for the pyruvate dehydrogenase complex always. Okay, what is it? What is this complex? This is a group of enzymes in the mitochondrial matrix. Okay, so in the mitochondria, as you can see here, this is the illustration. This is the outer membrane. This is the outer membrane. Okay, this is the inner membrane, and this is the mitochondrial matrix. Okay, this is the matrix of mitochondria. Okay, which is inside. Let me show you in this image. So this is the okay, this is the matrix of the mitochondria. This is also I can call this is the matrix of the mitochondria, okay, where the PDC complex is located, okay. So it's a group of enzymes. PDC is a group of enzymes in the mitochondrial matrix that prepares the pyruvate to enter to the Krebs cycle. It is just the preparation process for the pyruvate to enter to the Krebs cycle. Okay, where does the pyruvate came from actually? Okay, so pyruvate is made during the glycolysis in the cytoplasm, right? Each glucose gives 2 pyruvate molecules. So in order to make it happen, we need the oxygen. This PDC process only happens when the oxygen is available, when it's aerobic respiration is here. Okay, for example, if it's if in the hypoxia, which means low oxygen, this step cannot occur. Okay, so pyruvate is instead converted to the lactic acid I showed you, right? And it leads to the lactic acidosis in the intensive care unit patients. So what does the PDC do actually? Let's say pyruvate is a 3-carbon molecule. Do you remember the formula of the pyruvate? So pyruvate is a 3-carbon molecule. It has 3 carbons in the pyruvate, okay? This is first carbon, second, and the third carbon, okay? 3-carbon pyruvate, you can see here. So the function of the PDC is to remove 1 carbon, okay? Is to remove the 1 carbon. So here you see 1 carbon is removed. At the end, we are getting the acetyl-CoA. I am removing 1 carbon outside, as you can see, and 1 carbon is going outside as a carbon dioxide that releases the carbon dioxide outside, okay? The remaining 2 carbon unit is attached to the coenzyme A, okay? So it's attaching to the coenzyme A, okay? It's attaching to the coenzyme A, okay? And then it's making the acetyl, 2 carbon is called acetyl, and it's becoming acetyl-CoA, okay? So it's forming the acetyl-CoA. So from pyruvate, I am making the acetyl-CoA molecule, okay? So then NAD is reduced to the NADH. In this reaction, 1 molecule of NAD plus, which is oxidized state, which is ready to accept the electrons, can be reduced, will be reduced to the NADH, okay? So what are the products? What are the products of this reaction? So products is NADH production, right? So 1 molecule of NADH is produced, right? Acetyl-CoA is produced and carbon dioxide is produced, right? So from 1 pyruvate, but how many pyruvate I have? From 1 glucose, I had 2 pyruvate molecules, right? 2 pyruvate molecules. If I have 2 pyruvate to this reaction, I will have 2 NAD plus molecules as well and 2 coenzyme A enzymes, okay? As a result, the products will be 2 acetyl-CoA, 2 NADH, and 2 carbon dioxide at the end. Why 2, 2, 2? Because here it's just illustrated 1 pyruvate. Only 1 pyruvate is illustrated here. But how many pyruvate I have? From 1 glucose? I have 2 pyruvates, and 2 of them will go to the cytoplasm, from the cytoplasm to the matrix of the mitochondria, okay? That's why all the products will be 2, 2, 2. Here I just illustrated 1. If I put here 2, pyruvate is coming from 1 glucose, so coenzyme A will also be 2, okay? And 2 NADH will take part in this reaction. 2 carbon dioxide will be released, and 2 acetyl-CoA will be produced, and 2 NADH will be produced, okay? Or reduced, okay? So products from one pyruvate, products from the one pyruvate is 1 NADH, 1 acetyl-CoA, 1 carbon dioxide. Single 1 glucose gives 2 pyruvate, and you should multiply it into 2, and the products will be 2 NADH, 2 acetyl-CoA, and 2 carbon dioxide. Okay, so this is actually what you have to know for the you should know for the examination purposes. Okay, you know what's beriberi disease? Beriberi. Vitamin B1 deficiency. This is very vitamin B1 deficiency when the pyruvate dehydrogenase complex cofactor is missing. Okay, so what's the meaning of cofactor? I taught you during the first or second lecture, I don't remember. Cofactor is actually what makes the enzyme work. The example for the cofactor is vitamin. Okay, so I told you about the coenzyme, right? So coenzyme A, so this coenzyme should attach to the acetyl, right? So for this reaction, in order to happen this reaction, in order to PDC happen, PDC it's a complex of enzymes. Which enzymes? Coenzyme A's, right? So in order to this coenzyme work, I need a special vitamin which is called B1. The second name of the B1 is what? Which vitamin? Thiamine, right? Thiamine. So thiamine, we call it thiamine. So PDC cofactor is missing, so thiamine is missing, B1 is missing in the beriberi, less acetyl-CoA. What it leads to? It leads to the fatigue because less ATP is produced, and it leads to the confusion as well, okay, in the beriberi. So PDC deficiency, it can be genetic as well. Okay, PDC deficiency leads to the lactic acidosis because there is no oxygen, because oxygen is available, but PDC is not available. PDC is where we prepare the pyruvate for the Krebs cycle, okay? Especially in the newborns, its genetic cells cannot make acetyl-CoA. So acetyl-CoA is really important. We should make from pyruvate acetyl-CoA always, okay? We should make the acetyl-CoA because acetyl-CoA, it enters to the citric acid cycle, which we call the Krebs cycle. So pyruvate turns into the lactic acid instead. So it's very dangerous. So neurodevelopmental delay can occur in the newborns. Okay. Any questions? And the carbon dioxide you're exhaling from the lung. Do you see this carbon dioxide, which is releasing from pyruvate? Do you see this carbon dioxide? So carbon dioxide exhalation in the lungs comes partly from this step. Partly, I'm saying partly, comes partly from this step. It's measured in the ABG gas, arterial blood gases. It can be measured, okay? So I hope it was understandable, PDC complex.
Yeah.
These are the questions. So you have to do, you can do it on your own. Okay, please do these questions. And let's talk about the Krebs cycle. So what's Krebs cycle actually? What do you know about the Krebs cycle? Okay, Krebs cycle, this is repeating loop of the reactions. This is the repeating loop of the reactions inside the mitochondrial matrix. I showed you mitochondrial matrix, right? In before where the PDC is happening. So a Krebs cycle will also happen in the mitochondrial matrix, okay? It takes the acetyl-CoA. Can you see this acetyl-CoA? Where it came from? Where this acetyl-CoA came from? from pyruvate. Pyruvate became the acetyl-CoA, right? So pyruvate became the acetyl-CoA. So this is one acetyl-CoA. So acetyl-CoA, this is the illustration. This acetyl-CoA from the previous PDC step, right? From the previous pyruvate dehydrogenase complex, it's going to break down and it releases a lot of energy in the form of electron carrier. And it's called a cycle because it ends where it begins. Okay, it's going to end where it begins with a molecule which is called oxaloacetate, okay, which is oxaloacetate. So acetyl-CoA is going to mix with oxaloacetate and it's going to produce the citrate, okay, citrate. It's going to produce citrate in the matrix of the mitochondria, okay? So how it's all starting, acetyl-CoA, which has two carbon molecules, joins with oxaloacetate to form the citrate, okay? In the citrate, we have 6 molecules. You see in the oxaloacetate, you have 4 carbon. Here you have 2 carbon. And in the citrate, you have 6 carbon here, okay? This is where the cycle is going to begin. This is the end of the cycle. This is beginning of the cycle, okay? This is the beginning of the cycle, okay? So think of oxaloacetate as a reusable tool. It grabs the acetyl-CoA like a train engine picking up the passenger, drives them throughout the city, drops off the energy, and returns alone to the and it will be recycled again. So citrate is rearranged and broken down step by step. Okay, we are going to break down this citrate one by one. Okay, 2 carbons are removed and released as a carbon dioxide. Okay, I'm going to remove 2 carbon dioxide outside. Okay, as you can see here, this carbon dioxide is removed, this carbon dioxide is removed. Each carbon lost also triggers NAD plus to make NADH. Each carbon loss makes empty electrons. When you lose the carbon, one carbon lost is a carbon dioxide here, as you can see. When you lose carbon, so NAD plus will be reduced to the NADH. Okay, is it understandable? And then we have to make the energy from one acetyl-CoA. The cycle produces 3 NADH, 1 FAD, 1 ATP, and 2 carbon dioxide. Okay, this is for one acetyl-CoA. But remember, one glucose is going to produce 2 acetyl-CoA. So double it for one glucose. What are the results for the Krebs cycle? You just have to know the results. So from one acetyl-CoA, you see from one acetyl-CoA, you're going to make 1, 2, 3 NADH molecules. I'm going to make 3 NADH molecules. How many carbon dioxide is released in this reaction? 2 carbon dioxide is released, right? So 2 carbon dioxide is released in this reaction. How many FADH₂ is released from this reaction? 1 FADH₂ is going to release, okay? FADH₂, this is also electron carrier, okay? So 2 FADH₂ is going to be released in this reaction, okay? What about ATP? GTP and ATP, they're actually one thing. So energy carrying molecule equivalent to ATP, this is GTP. So 1 GTP is made. So 1 ATP is made in this reaction, okay? 1 ATP is made from 1 acetyl-CoA. This is from 1. This is for 1 acetyl-CoA, but I have 2 acetyl-CoA, right? Because I'm breaking down 2 glucose molecules. Do you remember? We obtained 2 acetyl-CoA in the PDC complex. From the PDC, we obtained 2 acetyl-CoA molecules, right? And I'm just showing you 1 acetyl-CoA here, but we have 2. If we have 2 acetyl-CoA, we will have 2 this kind of cycles. We will have 2 this kind of cycles. If we have 2 this kind of cycles, what will be the products? These products will be doubled. These products will be doubled. As you can see here, they will be twice as much. They will be twice as much, okay? So we will have 6 NADH, 2 FADH₂, 2 ATP, and 4 carbon dioxide released just from citric acid cycle, or we call it Krebs cycle, okay? So at the end of the cycle, the 4-carbon molecule oxaloacetate is reformed. Okay, it's ready to join another acetyl-CoA, and this cycle is going to repeat again. Okay. So what's going to happen? So carbon dioxide from the Krebs cycle measured in the arterial blood gases during the respiratory failure. Okay, if we have the respiratory failure, we're going to measure that released carbon dioxide from the Krebs cycle. Okay, in the thiamine B1 deficiency, we call it beriberi. This affects the enzymes in the Krebs cycle that leads to the confusion, memory issues, which we call the Wernicke's encephalopathy. Okay, and we can have the mitochondrial disorders, which impair the Krebs cycle, low ATP, which leads to the chronic fatigue and the muscle weakness. That's why the vitamins group B is really essential for the memory, for the ATP production. Okay, heart muscles, they runs always entirely on the Krebs plus electron transport chain, explains why cardiac ischemia leads to the immediate energy failure. Okay, so any questions so far from this topic? Think of acetyl-CoA as a fuel tank. Okay, acetyl-CoA like a fuel tank. The Krebs cycle is an engine always. Okay, the Krebs cycle is an engine, and acetyl-CoA is a fuel tank. It takes a fuel, I mean the acetyl-CoA, and burns it cleanly and produces electric power, mechanical power, and the smoke. Electric power is NADH and FADH. Mechanical power is actually ATP, and the smoke is carbon dioxide. This is like analogy, simple analogy to that. So this is the end of PDC and the Krebs cycle. So let's shift to the electron transport chain and oxidative phosphorylation, which is the last step of cellular respiration.